that blood flow is directly proportional to a pressure gradient, right? So the steeper the pressure gradient, or the larger the difference between the high pressure and the low pressure, the greater blood will flow. So it is a hydraulic system. Blood is flowing through channels, tubes, vessels, right? And that movement is driven by force. And the force is pressure being generated by the heart. We also know, right, that there is resistance to blood flow. And this resistance is governed by some basic principles of hydraulics, and the one that we really regulate to affect resistance is vessel diameter. If you do vasoconstriction, it means the radius gets smaller, the resistance goes up. And conversely, if you do vasodilation, the diameter is larger and the resistance goes down. So this is what we've been looking at here. Okay? And we talked about some factors that affect all of this. We looked at cardiac output as kind of, a driving thing for blood flow. We looked at resistance and studied all the different factors about that. But what we were kind of ended on last time on Friday was a discussion of how does the heart generate this pressure, right? So we were looking really at pressure from the standpoint of the heart. Now, when we talk about blood pressure, what we're really measuring is arterial blood pressure, pressure out in the arteries. That is determined, though, right, by pressure generated by the heart, in particular by the ventricles, and since we're talking about systemic blood pressure, by the left ventricle. So when the left ventricle contracts, the force with which it contracts is what determines your arterial blood pressure. And that determines blood flow, and that determines exchange of capillaries, as we'll see today as well. Okay. So when we look at the heart, if you remember from anatomy, right, there was an epicardium, an endocardium, and a myocardium. Right? And the myocardium was the layer of the heart that had the muscle tissue in it. Right? And the muscle tissue that it had in it was cardiac muscle tissue. Okay. Now, the different tissue types, just are summarized kind of here. What we did, we spent time talking a lot about skeletal muscle tissue. We talked a little bit about smooth muscle tissue. But by comparison, right, remember cardiac muscle tissue is striated. By saying it's striated, what that means is it has sarcomeres. That arrangement of actin and myosin is what gives it those striations. The tissue, the cells tend to branch, and they have intercalated discs. And the things about the intercalated discs is that those are modified gap junctions, so those are electrical synapses. So if one cell gets a signal, it's going to spread rapidly to neighboring cells. So that's what that means by that. The control of contraction, again, calcium is the ion that controls it, just like the other two. And like skeletal muscle, there is a troponin-tropomyosin complex. So it's the binding of calcium to troponin that will initiate that power stroke cycle. Okay? That source of that calcium, interestingly enough, right, is that some of that calcium, like smooth muscle, comes from extracellular sources. We'll start in a minute. We talked about this excitation-contraction coupling in these contractile cells. There is some extracellular calcium that comes in. Okay. There are also in cardiac muscle tissue specialized cells called autorhythmic cells, whose job it is to generate action potentials. So because of that, we say that the contraction of cardiac muscle tissue is autorhythmic. However, it is modulated by autonomic nerves from the CNS, meaning that when the heart rate needs to increase or decrease from some set point, it is your sympathetic and parasympathetic branches of the autonomic nervous system that allow for a heart rate change. So that's what we've been looking at. So if you remember from anatomy and in our discussion Friday, we said that there are two different types of cells in cardiac muscle tissue, so myocardial cells. The bulk of them are contractile, and these are the ones that generate force. Okay? There are localized to only two places, the SA node in the superior part of the right atrium and the AV node in the inferior part of the right atrium that contain these clusters of autorhythmic cells. And those are at nodes, right? The SA node and the AV node. And what the autorhythmic cells do, now they are muscle cells developmentally, but they're modified. They don't have sarcomeres, they don't have actin and myosin, so no contractile elements at all. In fact, all they function in is generating action potentials. Okay? So these are the cells that are responsible for telling these cells to contract. And when these cells fire an action potential, it will spread to these cells and they'll contract in response to that. And we saw that we can look at that electrical activity that's happening and coordinate it with the mechanical activity by studying the events of the cardiac cycle. And we looked at the EKG tracing and we labeled it and we said this is when atria depolarize and contract, ventricles depolarize and contract, and so forth. Right? And that's just in response to these autorhythmic cells initiating all of that events of the cardiac cycle. Now, these cells, as we'll see in a few minutes, it's kind of cool how they work, will spontaneously depolarize at a rate of about 100 beats per minute. So at about 100 times every minute, these cells will spontaneously generate an action potential. Now, if that was transmitted at 100 beats per minute or 100 action potentials per minute to these contractile cells, your heart rate would be 100 beats per minute. Now, clearly that's too fast for at rest, so we know we can modulate that signal. So we can slow that signal down, and that would be the actions of your parasympathetic branch of your autonomic nervous system. And then if we don't do anything, the heart rate's 100, but sometimes as part of a sympathetic response, the heart rate even goes above 100. So that would be from sympathetic stimulation. So this dual innervation, parasympathetic and sympathetic, is used to regulate the heart rate. At rest, it's parasympathetic tone, constantly saying slow down, slow down, slow down, slow down, right? And then in response to an activity, we either remove the parasympathetic or increase the sympathetic to change the heart rate.